Hello everyone, this is your instructor Kwame Shen. So this lecture is a first part of the lecture on microbial growth and control. And so this component will concentrate on just the microbial nutrition and growth, and the second part would then focus on microbial control. So for microbial nutrition and growth, the objectives will be to look at the requirements that these microorganisms need for growth, the sources of their food, the sort of chemicals that they need, and the environmental factors that influence their growth. We'll also look at some associations. In associations, specifically, we'll look at biofilms, and then laboratory settings, in terms of the basics when it comes to culturing microorganisms, the different media, and the different attributes that they have, and therefore how we employ them in the lab. And then in terms of the growth, how bacteria grow, different terminologies that we employ in describing bacterial growth, the different phases, and how we can measure bacterial growth. So generally, when we talk about growth in living organisms, we tend to focus on the size. However, when it comes to microbial organisms, when we talk about growth, we focus more on the increase in numbers, of the microorganisms. And microbial growth on surfaces may be in the form of colonies. So if you're talking about colonies, you're talking about bacteria that all originate from a single cell. We typically say colony forming units. So on this plate, these are bacterial colonies. But more generally, bacteria will exist as biofilms. There's an example of a biofilm, and that's an example of a biofilm. So these are groups of bacteria that are attached to each other, and this was found on the toothbrush bristle. The same with this as well at a higher magnification. Bacterial growth in liquid would lead to turbidity. So if you look at these two samples here, this looks very clear and this looks more opaque because of the bacterial growth. We characterize microorganisms based on the source of their carbon and how they generate their energy, how they generate ATP. So with this criteria, we can group organisms in terms of prokaryotes or bacteria into autotrophs and heterotrophs. Autotrophs are those that will use carbon dioxide, inorganic carbon dioxide, as a source of their carbon. The heterotrophs, on the other hand, will use organic compounds. So organisms that have already used inorganic carbon dioxide as their source of carbon, the food that they produce, the heterotrophs will then rely on those. Now, we can break these groups further into photoautotrophs and chemoautotrophs. The photoautotrophs will use light as a source of energy. The chemo will use chemicals as a source of energy. The same way we can group the heterotrophs, which rely on organic compounds as a source of carbon, into photoheterotrophs and chemoautotrophs. Photoautotrophs will use light, chemoautotrophs will use chemicals as their source of energy. So autotrophs will produce their own food by obtaining carbon from inorganic substances. Heterotrophs cannot synthesize their own food, so they have to depend on other organisms for their source of carbon. In terms of how the energy is produced, the phototrophs would capture protons in order to acquire the energy. And the capturing of the protons is caused by the excitement of electrons caused by the sun's rays. The chemotrophs, however, would obtain energy by oxidizing an electron donor. So they're going to accept the electron and a hydrogen atom from another molecule, and in the process they're able to create their energy. So if we look at photoautotrophs, we see them in the oceans. We have cyanobacteria in algae, chemoautotrophs as well, in the deep seas. Example is sulfolobus, and deeply in the soil as well. Example will be the nitrogen fixing bacterium, rhizobium is an example. Then we have photoheterotrophs. Example would be chloroflexus, and so these are thermophiles, so they live in hot environment and hot springs. And most bacteria, however, will fall under the chemoheterotrophs, where they will use energy from chemical reactions to generate their ATP, and also rely on organic compounds from other microorganisms as their source of carbon. So this table summarizes all that I'm talking about, where you have phototrophs using light as a source of energy, CO2 as carbon source, and the examples of organisms that carry out metabolic processes this way. And then we have anabolic processes rather, because they are building. Then we have chemoautotrophs that will use energy source in terms of inorganic chemicals, and then carbon source will be CO2. An example will be the sulfolobus. Then you have the heterotrophs that will use light, source of energy. This photo will use the light, the chemo will use organic compounds, but they all will use organic compounds as a source of their carbon. What then do these microorganisms require in terms of enzymes, in terms of vitamins that they need? So we group organisms based on how they react to oxygen, which we call, these are reactive oxygen species, that if the organism doesn't have certain chemicals in terms of enzymes, these reactive oxygens or energized oxygen species may destroy them. So based on that, we group organisms into aerobes, obligate anaerobes, facultative anaerobes, aerotolerant, and microaerophils. Now the aerobes will require oxygen. So they have a terminal electron acceptor being oxygen. An example is pseudomonads. That's pseudomonas, the picture with all the flagellum. And then you have obligate anaerobes. They require a final electron acceptor, but this electron acceptor would not be oxygen. So other than oxygen. So example would be sulfur or hydrogen sulfide, nitrates, and other inorganic compounds like that. Then you have, because for these obligate anaerobes, oxygen is poisonous to them. An example would be Clostridium tetanae, that causes tetanus. Clostridium botulinum, that causes botulism. So these are some of the examples of organisms that are obligate anaerobes. Then you have facultative anaerobes. Facultative anaerobes would grow both in aerobic and anaerobic environments. However, they are more efficient when oxygen is around. So when they have access to oxygen, they are more efficient. Without oxygen, they will grow, but at a slower rate. An example is E. coli. Then you have aerotolerance. They will grow anaerobically or aerobically. They are indifferent to oxygen, and so they have enzymes that help them to survive in the presence of oxygen. Then you have the microaerophils. They will grow aerobically, but they need low concentration of oxygen. An example will be Helicobacter pylori or H. pylori that exists in the stomachs. So if we look at oxygen concentration, based on oxygen concentration, we can tell how these organisms will spread out in a tube. So if we look at obligate aerobes, obligate aerobes would form closer to the cap because that's where the most oxygen is. They will be in the area where there is most oxygen present in the media. For facultative anaerobes, they would also be closer to, in terms of concentration, highly concentrated where there is most oxygen, but they will be found throughout the media. For aerotolerant, because they are indifferent to oxygen, they will be spread evenly throughout the media. Strict anaerobes would be way away, far away from where there is more oxygen, and they will be concentrated where there is less oxygen, which is towards the bottom of the tube. Then you have microaerophils. They wouldn't be closer to the top. They'll be just beneath it a little bit because they need just enough oxygen, but not too much of it. Oxygen is a poisonous species that living organisms have to deal with. And in this regard, we're speaking especially of reactive oxygen species. Oxygen that has taken on extra electrons and that's making it unstable, and so it destabilizes other molecules that are close by. And so we'll talk about 4 of these reactive oxygen species. We have singlets, O2 minus, and these are regular molecular oxygen that is at an excited state, a higher energy level, which makes it unstable. And organisms that have these species in them have to find a way of dealing with them. Examples would be the phototrophs, these plants that produce fruits, and they have all this coloration, which pigments, carotenoids, which help protect them. Then you have superoxide radical. Superoxide radicals, this is their configuration. You see the extra electron. They have organisms that produce these radical species would have to have an enzyme, superoxide dismutase, in order to deal with them. We then also have peroxide anions, O2 minus, and for the O2 minus, that's the configuration, and they are produced from the remainders of superoxide dismutase. So you have hydrogen peroxide, which is produced from that reaction from organisms that deal with these reactive species. You also have hydroxyl radicals, OH minus, and you see the extra electron, and it's formed from incomplete reduction of hydrogen peroxide. So in more detail, you would have singlet species that damage and are reactive to organic molecules, especially plants that have chlorophyll, because they use the sun's rays and have to trap it. And so they have a protection. They have these pigments, carotenoids, that help protect them. For organisms that produce the superoxide radical O2 minus, they have to synthesize the enzyme superoxide dismutase, which will convert these radical oxygen to hydrogen peroxide, which is less toxic. It is still toxic, but it's less toxic. For the peroxide anions, O2 minus, the organisms that produce them, it is still toxic to them. They have hydrogen peroxide, which they have to catalyze into water. And these organisms will have the enzyme catalase. So when you do biochemistry in lab, you would come across organisms that are catalase positive and those that are catalase negative. So pay attention to that, especially Staph aureus. Then you have peroxidases. The peroxidases would also convert hydrogen peroxide to just water. The difference is that catalase will convert hydrogen peroxide to water and oxygen, and that with those reactions you see the bubbling action, the effervescence that's being produced. And then for peroxidases, it is just water. Then you have the hydroxyl radicals, OH minus, which is an intermediate form of oxygen, and it's formed in small amounts for even regular organisms, normal respiration, but they don't last, and so they're very temporary in their existence. And for organisms that produce hydroxyl radicals, you still need SODs, superoxide dismutase, and you need other catalase or peroxidases to convert the hydrogen peroxide to water. So remember, these are the enzymes that you need to pay attention to, and the organisms that produce them as well. So that's an important slide. You have obligate aerobes. They need superoxide dismutase. They have to deal with oxygen. And then they also have catalase that would have to convert the hydrogen peroxide into a more tolerable form, which is water and oxygen. It's the same for facultative anaerobes. They have SOD and catalase. For aerotolerant anaerobes, because they are more or less intolerant, they are indifferent, to oxygen species, and they only have one enzyme, the SOD, which will convert the reactive oxygen species to hydrogen peroxide. For obligate anaerobes, they do not have any of the enzymes, and so if they are raised or cultured in the presence of oxygen, they will die because they have no way of dealing with a reactive oxygen species. Then you have microaerophils. They will need a very little amount of oxygen, so all of them will have SOD, and some will have catalase and others will not. Microorganisms, just like microorganisms, in this regard, prokaryotes, just like eukaryotic organisms like ourselves, we need other chemicals as well to survive. They will need nitrogen, they will need sulfur, they will need phosphorus as well. They will need nitrogen for their protein synthesis, for their nucleotide bases, and they will use them in the form of nitrates or ammonium. And then some of them are able to fix atmospheric nitrogen and make it useful for all microorganisms and other living organisms as well. An example of a microorganism that does that is the rhizobium species. Sulfur and phosphorus. You need sulfur for protein synthesis and the synthesis of vitamins. And vitamins would act mostly as coenzymes in reactions. And then phosphorus is needed in the synthesis of nucleic acid, the plasma membrane for phospholipid layer, and ATP. Then you have trace elements. So for the trace elements, you're talking about elements or minerals that are needed in very tiny amounts. So calcium and magnesium and copper and the likes, which are also cofactors in biological systems. So there's a difference between cofactors and coenzymes. They're cofactors if they're inorganic in origin. Okay. The main chemicals are hydrogen, oxygen, nitrogen, and carbon. And then you have sulfur and phosphorus as well. What then do these microorganisms require for growth? We've seen their general characteristics. We've seen the tools that they have in terms of the enzymes that they're able to produce or not, and the different conditions that they can survive. Now, for physical factors that affect microbial growth, you have osmolarity, temperature, and pH. For temperature, you have an optimum temperature. That is the best temperature at which they will grow. And for most microbes, it's around 37 degrees Celsius. There is a minimum growth temperature and there is a maximum growth temperature as well. Now, if temperature is too high, it denatures the 3D nature, the protein, and therefore renders the protein dysfunctional. Then you have, in terms of temperature as well, there is an effect of temperature on the plasma membrane. Remember, the plasma membrane has a triglycerol, so it has components of glycerol and fatty acids. The fatty acid will be a combination of saturated and unsaturated fatty acids. So because of that combination, the temperature is important. If it's too high a temperature, it's going to loosen up the plasma membrane beyond a certain temperature and therefore render the plasma membrane ineffective as well. It's going to lose its integrity. For temperature, in terms of minimum growth, optimum growth, and maximum growth, if you look at these three plates here, so this is around the minimum growth at which you see temperature at which you see growth, 22 degrees Celsius. You still see growth at 30 degrees Celsius. It's more than 22, but you see the most growth at 37 degrees Celsius. So this is the optimum temperature. If you graph it at optimum temperature, you see the highest growth rate. Okay. Based on the temperature that microorganisms prefer, or what is best for them, their optimum temperatures, we can group microorganisms. And so we have the psychrophiles, and the psychrophiles, they are cold loving. They love very cold environments. So they will grow at temperatures that are around 0 degrees or even sometimes below. We have another group, the psychotrophs. The psychotrophs would grow in cold temperature, but not as cold as the psychrophiles. They used to be described together, but recently they've been split up. And so the psychotrophs, which you can see here in this graph, the psychotrophs would grow in temperatures that is around 0, but all the way to around 30 degrees they will still grow. The optimum temperature is around 15 to 20. And these are the microorganisms that cause food spoilage, because we tend to store our food in cold temperatures, not in freezing temperatures, but just in cold temperatures in the refrigerator. And so these microbes are food spoilage microbes. They will slowly degrade the nutritional contents of the food and cause it to go bad over time. Then you have the mesophils. They love moderate temperatures. Most microbes are in this group. They will grow optimally around 37 degrees Celsius. You have the thermophils. They have the heat-loving microbes. They cannot grow below 45 degrees Celsius. So there has to be more heat. And then you have the hyperthermophils, extreme heat. So we've seen some organisms grow around 121 degrees Celsius, and these are the archaea bacteria, the hyperthermophils. Then you have pH. So for pH, it's either acidic or basic or neutral. Most organisms will like a pH that is around a neutral pH. However, and that in the presence of very acidic or basic pH, they become denatured. So it's the same principle as the effect of heat on the enzymes that these microorganisms use. However, you have microorganisms that actually would want or would love or would tolerate an acidic environment. An example would be the Helicobacter pylori. You have others that would like an alkaline environment. We call them alkalinophiles. They love alkaline environment. An example would be Pseudomonas. Then you have osmotic pressure. So microorganisms would obtain their nutrient from water. So the water in the environment, you have the food particles or the nutrient that will dissolve in the water, and then they would then be able to absorb it through their membrane, through one of the transport means. So generally, you have microorganisms composed about 80 to 90% water. You have about 0.85% salt, so sodium chloride content in microbial cells generally. There are exceptions. And so whatever environment that they're in, the concentration of water in there as compared to salt has an effect on them. So if you place a normal cell in an isotonic solution, you don't expect any change in terms of the effect of water. The net flow of water in and out of the cell is the same. If you place a normal cell in a hypertonic solution, so the cell has a sodium chloride concentration of 0.08%, then you have an environment that has a sodium chloride concentration of 10%. There is more salt here and less water, so water is going to diffuse by osmosis from the cell to the environment. It's going to result in plasmolysis. Okay, the cell is going to shrink. And then if you place a cell in a hypotonic solution where you have a concentration of 0.3%, the cell has 0.85%, water is higher in the environment than in the cell, it's going to move towards the cell, into the cell, and eventually cause the cell to burst. So all these effects can be experienced by microorganisms. Osmotic pressure then is an important effect on the growth of microorganisms. The exceptions that we have would be, for instance, the halophiles or the obligates, the extreme or obligate halophiles, and the facultative halophiles. So these organisms would either tolerate or would need a very saline environment, very salty environment in order to survive. Microorganisms may exist singularly, like a colony on its own, but more generally, microorganisms exist in communities. That is, they attach, it starts with a free-swimming phytoplankton that attaches to another one, and so they attach to each other until it gets to a point where there is enough of them to give a signal, and produce some chemicals that communicate to other bacteria to come to that area where those initial ones are aggregated. And so you have more and more of them aggregating in that area, and there's more chemicals that are synthesized. Now, the minimum bacteria that you need for this process to be invoked is known as the quorum. And so the effect that they produce is known as quorum sensing. So quorum sensing is a form of cell-to-cell communication. It regulates gene expression among the bacteria biofilm, and they tend to behave like multicellular organisms in such a state. Really, really, really fascinating. Very, very fascinating. And then when they come together like that, they will then synthesize chemicals called inducers, and these inducers will then attract other bacteria to come to that area, and more of them will come. And when they come together like that, they're able to form a community that gives them protection. They're able to even sometimes hunt for food as a group, just like multicellular organisms. And in terms of healthcare, microorganisms that form biofilms, and it could be a combination of either different kinds of microorganisms or the same species of organisms, but they are together. They are very difficult to treat in hospital settings, so they account for a number of healthcare infections, and these infections are very difficult to treat, mainly because these microorganisms exist in a group like that, so they're able to protect each other. It begins with a single floating bacteria, and then other cells will aggregate, and then they get to a point where they're able to induce others to come, and then they form this colony and get to a point some of them will break off, and then they'll go and start a new colony again. Very, very, very fascinating. How then do we grow microorganisms? We grow them in the lab, and that's known as culturing, giving them the right environment for them to be able to grow. We supply them with nutrients, and it's in the form of a media, a culture media. The culture media may be a solid. If it's solid, we'll have it in a plate, so you may have it in a tube in the form of a slant, and that's an agar. Or it could be a liquid, and if it's a liquid, it's called a broth. If you're culturing a microorganism, you introduce a small amount of the microorganism onto the culture. And that amount that you introduce is called the inoculum. That's the inoculum. You could describe bacterial colony differently. You could describe them as circular or irregular. You could describe them based on the edges. Is it entire? Is it undulates? Is it filiform? And all that. Or you could describe it based on the elevation. Is it raised? Is it convex? Is it flat? So those are some of the ways you can describe a colony. And then the culture that grows, you have a pure culture. A pure culture is made up of the same species of organisms. So it's not a mixed culture. A mixed culture may have two or more organisms together on one media. You could have plates, so these are plates, and we streak these plates. We call it streaking for isolation. So you streak so that you end up with single colonies like that. So you have the first streaking, second, and the third, and the fourth, and as you're going, you're reducing the amount of organisms that you're taking to the next quadrant. So that finally, the last quadrant, most likely you would have isolated colonies. So that's an isolated colonies. Okay. The colonies that you have here are coming from a single cell. So it's a single cell that has produced them. And so we describe them as a colony forming unit. This is not just one cell, it's a number of cells. And this is more zoomed in, and so you can see the colonies more clearly there. You require a septic technique, so this media has to be sterile. The loops that you use to transmit the organism, the inoculum, has to be sterilized. You have to have gloves on, and the environment has to be sterile. The stock, wherever you're transferring the organism from, has to be sterile as well. If it's a tube, every now and then, when you remove the organism, before you remove it, you have to flame it, and then after you remove, you have to flame it as well. I think these would be elaborated more in the lab section of this course. We have different media that we use, and it depends on what we are looking for. Some media are selective, so they have specific nutrient that will only allow certain organisms to grow. A lot of the prokaryotes that exist have not been cultured yet, so we don't know how they appear on media. For media, one component that we rely on heavily is agar. Now, agar is a complex polysaccharide that is found in the cell walls of red algae, the red algae. Microbes are not able to digest them, so they are a perfect scaffold. They are a perfect scaffold, or a base in which we can actually infuse these nutrients that we want the microbes to feed on. The good aspect of it also is that they don't dissolve until at 100 degrees. So even the thermophiles, not extreme thermophiles, but the thermophiles, which would need temperatures between 50 and 60 degrees, can still be cultured on them. And then they will solidify below 45 degrees. So it is safe to melt them, add the additives to it, and then at room temperature, at 25 degrees or so, they would be in the solid state, so that you can actually apply the microorganisms on them, and then incubate them. Perfect, very perfect for our needs in this regard. This table summarizes the different types of media. You have the defined media. A defined media is a media that you know the exact components of the media. So the nutrients that are in there, you know the composition of the nutrients that are in there. There are instances where we use yeast or beef or soy, like tryptic soy agar, which is a very common agar, that general purpose agar that we use in the lab, or milk may be the source of some of these agar. And if that's the case, we don't know the exact composition. We know the source, but we know the exact composition. And so that's an example of a complex media. Then you have selective media, which will have components that will favor the growth of some organisms but not others. You have differential media. Differential media usually will have some component that will show a change in color. So you have blood agar, which will show different colors based on the organisms that are going on them, whether it's an alpha, a beta, or a gamma hemolysis. You also have anaerobic media, and these are media that is free of oxygen. Generally, you would actually have a setup where a candle is added to the setup, and so that it sucks the oxygen and produces more carbon dioxide, which the bacteria may need to grow. So these are examples of differential media. This is a differential media. There's a blood agar differential media. This is McConkey agar. It's both selective and differential. So you see E. coli, it presents differently. It metabolizes the nutrient differently than Staphylococcus aureus, which doesn't even grow at all. So in this case, this is selective. And then if you look at E. coli and Salmonella enterica, you would see that Salmonella grows differently in terms of how it presents, the coloration, and E. coli presents differently as well. So in this case, it's a differential media. So there are some media that is both selective and differential. An example is McConkey agar. In addition to the media that I've talked about, you have animal cells that are used in growing viruses mainly, mycobacteria as well, and Treponema pallidum, which causes syphilis. You have low oxygen environments for microtolerant and microaerophils, aerotolerant and microaerophils, which I've described earlier, that its setup may include candles. And then you have enrichment media, where you have an added component to it. An example of an enriched media is blood agar. There are instances where you could have a cold enrichment as well. For methods of preservation, if you want a short-term storage, refrigeration, because it slows metabolism. If you do deep freezing, it's going to stop metabolism. That's for long-term storage. And lyophilization, which is a removal of water. Water is sucked out, is sublimed, and that in those instances, the products can be kept for a longer period. So now we'll look at the growth curve of bacteria, how bacteria grow. They grow by binary fission, so a cell divides and divides and divides and divides. First of all, the nucleic acid components, the DNA, will replicate. The cell then will become longer, preparing the cell for division. The nucleic acid will divide, and then the cytoplasm and the cell wall will then divide as well. Now, bacteria have what we call a generation time. A generation time is the time it takes for one bacteria to divide into two. So you have a single bacteria. The time it takes for it to go from here to here is the generation time. The time it takes to create the next generation. Okay. So if you look at generation time, For E. coli, this is the generation time for E. coli. E. coli divides every 20 minutes. So E. coli with the right conditions, with the right food supply and all that, every 20 minutes it's going to divide. So you have, in a period of 0, you just have 1. After 5 generations, you're going to have 32 of them. After 10 generations, you're going to have 1024. So 2 raised to the power the number of generations will give you the number of cells. By 20 generations, which is 400 minutes, you have over 1 million of them. I mean, just imagine. It's different for different microorganisms. Some of them may take hours, some of them may take days, but generally it's a very quick process for microbes. Then the growth follows a certain pattern. There is a phase. There is a lag phase where it's on the surface as if nothing is happening, but the reality is that at this stage, the cells of the microorganism are preparing for division. They're synthesizing the enzymes that they need. They're synthesizing the other molecules that have to come together to form the new structures that they are about to produce. Then you have the log or exponential phase. That is where you have active division of bacterial cells and growth. And it is the stage where you have the highest cellular respiration taking place. So cellular respiration is taking place, high metabolism is taking place, and high anabolism is taking place as well. So a lot of activities, full of activities. And this is how it goes. At a certain point, you have an equal, a minimum amount of cells that are produced constantly. At a constant rate, you have a minimum amount of cells that are produced. So you have this exponential graph that shows in terms of number of cells over time. It gets to a point where this stops. The point at which the log phase stops is known as the carrying capacity of the bacteria for that environment. The carrying capacity is the amount of space or resources in terms of nutrients that is available that can support the growth of the bacteria. So at this stage, this is the highest amount in terms of number of cells or bacteria that this environment can support. That's the carrying capacity. At that stage, the growth becomes stationary. So the rate at which the organisms are dying and the rate at which they are being produced is the same. So cell death is equal to cell renewal. The nutrients is depleting until they get to a point where the rate at which they are dying will be higher than the rate at which they are being replaced. And that's the death phase or the decline phase. It's important that you understand these phases and understand what's going on at each of these phases as well. How then do we measure bacterial growth? Now that we have a better understanding of the various environmental factors that influences them, their own chemicals, enzymes that they have, their needs, and how they grow. How do we measure growth? We can do growth measurement directly with incubation. So you have plates, you incubate, or you filter, or you find the most probable number, or without incubation, doing microscopic count or direct electronic counts. Then you could do it indirectly using turbidity, which is the most commonly used. You could also use metabolic activity. You measure a population's metabolic activity based on the amount of nutrients that they are metabolizing and the products that they are producing. And then you could also look at the dry weight, especially for microbes that are filamentous. So you could actually dry them and weigh them. You could do broth cultures, and then you could also do PCR as a way of measuring the content of microorganisms in terms of genetic composition. For the direct methods, and in specifically microscopic counts, so you have a specialized mounted support in terms of the glass slide that you use. You need about 10 million organisms per milliliter to do the direct count. So that's one limitation. And you have a glass cover, and then you put it in a microscope, and then you observe to see the number of counts in terms of bacteria that you see within a particular quadrant. So each quadrant is 1 millimeter cube. Now, what this does is that you are looking at the number of viable cells, and that's one assumption. And a viable cell is a cell that is able to reproduce, grow, and divide and produce a new colony. So that's one assumption. It's not a good assumption because not all cells would grow and divide. Because bacteria exist as chains and clumps and pairs, they don't always exist individually, so they can divide and then grow. And then you also have a way of estimating. So in terms of the good aspect of it, you can actually estimate the number of cells pretty easily. What you need to do is to count the number of cells that you see under the microscope. So you count the number of cells. You could count for one and multiply by the number of squares that you have in a quadrant that gives you. And then you would divide it by the total area in terms of volume, okay, of the area that is counted. And that will give you the number of cells per milliliter in that particular sample. You could also count the number of colonies on a plate and determine the number of bacteria in a sample by doing what we call direct plate counting using serial dilutions. It's a very commonly used. You will talk about this in the lab as well. So with serial dilutions, what happens is that you would have an original culture sample. So let's say you have 1 milliliter of that. You introduce 1 part of it into 9 parts of water. So this 1 milliliter is then added to 9 milliliter of water. So that's a 1 to 10 dilution factor. And the reason you do this is that you could as well culture all these bacteria on a plate and try to count them, but there will be millions and millions of colonies. You will not be able to count them. So what you're doing is you are diluting the amount of organism in a very regularized way, so that you can follow and be able to do the multiplication and find the number of organisms that you have in your original sample. So this is done serially until you have 1:100,000 dilution factor, and then you plate 0.1 milliliter of all that on a sterile plate. And then you incubate them and then count the number of colonies. Now you get to a point where there will be no colonies. So that's the concentration beyond which you wouldn't find any bacterial growth. And then you'll find where you have the minimal growth. Now, the minimal that we had in this example is 6 colonies. Then you have for 1:1,000, you have 65, and on and on and on. So these are 2 numerals to count. So TNTC, 2 numerals to count, 2 numerals to count. So you can actually count them. But we can use this to estimate the number of cells in the original sample. And how you do that is that you count the number of colonies on the plate, in this case 6, and then you multiply it by the reciprocal of the dilution sample. So the reciprocal of the dilution sample is 1/10,000. Reciprocal of it will be 10,000. So it will be 6 times 10,000. And that's going to give us 60,000 bacteria cells that was in the original sample. So that's how you estimate that. And that's something you should know as well. Understand how it works. Then you have direct methods, filtration, where you filter using a membrane. So vacuum is attached. You have a membrane attached here, and filter through here. You need about 100 milliliter of water to do that, and it's mainly used to check fecal contamination in water bodies. So you filter it and then you transfer the filter to a plate and incubate. So if you see colonies on there, it means that you have some bacteria sample in there. Another direct count method that is done is known as the most probable number, and it's more of a statistical method. So you have dilutions as well. So this is 1 milliliter. It is then diluted 1 to 10 factor and then 1 to 100 factor. Then you have tubes that you're going to inoculate these bacteria with. So you have first a series of 5 tubes. You inoculate this with, and these are the diluted sample weight, and this as well. And what it does is that all the 5 are incubated, and then you check for the number of positives. So for the undiluted, you have, say, 4 positives. The one that has a dilution factor of 1 to 10, you have 2 positives. And one with 1 to 100, you have, say, 1 positive. And then you would then count the number of positives. So it's 4 is to 2 is to 1. There is a most probable number table, that's a standard table, that you would compare it to and then determine the most probable number of bacteria that is present in the original sample. Now, if you do the 4-to-1 combination against the standardized table, you get around 26 cells of bacteria. So there is a range, but what the most probable number tells you is that there is a 95% chance that the number of bacteria that is in that sample is this amount. It gives you a range, it's between this range, but the most probable number is 26 in the example that I cited. And then you have indirect methods where you have electronic counts. And so you have a sample, you have an instrument that you just put it in, and it electronically counts the number of bacteria in there. Most laboratories use this method. Then you have the turbidity method, and it operates on this basic concept that a sample that has growth in it will be more turbid than a sample that has no growth. Now, if a sample that has growth in it is more turbid, it means that it's going to influence the amount of light that passes through, that is able to be transmitted through it, and the amount of light that is absorbed by the cells. And the two will run in opposite directions, inversely proportional. So, for instance, you have a blank tube without any sample growing in it. Light will pass through directly. So in terms of absorbance, you have 0 absorbance. In terms of transmission, you have 100% transmission. But once you have bacteria suspended in there, the transmission is not going to be 100, it's going to be less, and then the absorbance is going to be higher. So absorbance runs in this direction, transmission runs. And so you use a spectral photometer to measure the turbidity. So that's another way of measuring. So this brings us to the end of the first part of our lecture on microbial growth and control. This was mainly on microbial growth.